When you need to make nice figures to visualize gene expression profiles, but you are not expert in programming languages such as R or Python. Or sometimes you may need to quickly understand the expression pattern distributed across different datasets or samples. PlantGenIE expression tools will make this easy for you. In this screencast, I am going to show you how to quickly investigate the gene expression profiles. All you need to do is having some interested genes in your gene list. If you don't have genes in your gene list, please watch the previous video to add some genes into your gene list. I assume you already have interested genes in your gene list. Now you need to navigate to the PlantGenIE exImage tool. The exImage tool can be found in this URL. Once you are in the tool, you may see the different samples shaded in different colors. These colors are based on expression values that we calculated from RNA-seq pipeline. If you are unfamiliar with RNA-seq pipeline, please check the following URL to read more about the pipeline and understand how to calculate expression values. We can see there are two modes of visualizing exImage data. One is absolute mode and other one is relative mode. Absolute mode uses plain VST values. VST means variant stabilization transformation values that we generated from RNA-seq pipeline. Relative mode values were calculated using relative mean difference. Let me explain what relative mean difference is. Let's assume we generate a listed expression values shown in following samples: shoots, roots, young leaves, mature leaves, flowers. Then we calculate the average VST values. In order to calculate the relative values, we need to subtract average value from the absolute VST values. In other words, relative values depend on the expression patterns across number of samples for a given gene. We use relative values to amplify the expression differences and highlight significant color changes between samples. Unlike the absolute expression values, relative values cannot be compared with the expression values in another gene. Relative values is a ratio. There is no unit associated with the relative values. As you see, there are several RNA-seq datasets. In other words, experiments are available in PlantGenIE exImage tool. Here you can choose different experiments by using different select options. Finally, the user can download the publication ready figure as a PDF or SVG using corresponding export buttons. Thanks for watching. Next video I am going to show you another expression tool called explode.